From pharmaceuticals such as morphine to high-value ingredients used in foods and cosmetics, many plant compounds remain stubbornly resistant to production through microbial fermentation. But Fermelanta reckons it might have cracked the code.
I think what the biomanufacturing process really enables us to do is to get access to these really rare ingredients that were previously not available or available in such small quantities that we just can't use them efficiently at scale, right? So what we do is we really have the process down to enable companies to make ingredients that are already existing more efficiently and more cheaply, but also enter new markets that just weren't available before by using this technology.
So Fermelanta has reproduced the enzymatic pathways to produce lots of these molecules in a microbial system by introducing an unprecedented number of plant genes into a single bacterial cell. I mean, how many plant genes are we talking about here?
Yeah, more than 20, almost 30 genes into a single strain. And really the benefits of being able to introduce so many genes is that we have a single fermentation step to produce those. If you would break those down into several fermentation steps, it wouldn't be as efficient.
Yes.
But for us being able to introduce the amount of genes, we really can streamline the production and access molecules of a complexity that just weren't able to produce before.
So you are able to make phytochemicals in a microbial system that as far as you know, other companies are not able to do at the moment.
Yes. That's introduction of dozens of genes are key because we have the proprietary, unique, our proprietary tool to efficiently introduce that kind of number of genes. And we don't use that maybe patented tools like CRISPR. And so that's our advantages. And that means maybe we can, the current technology, existing technology is just make like simple molecules or proteins that require only one or few genes, just it. And so like we can expand this, the more pipelines or more target, and that's our what we can do, yes.
Traditionally, where our company came from was from the essential medicine space. So really what our founders have been working on for the last 20 years was making strains that can produce these rare ingredients for essential medicines like morphine. But since then, we have also seen a huge interest of rare ingredients in other markets. So not only in the essential medicine space, but we're now expanding into rare flavors, rare fragrances, or supplement ingredients where they also see a need for natural solutions, but the high costs are holding them back. So internally, we're mostly working on ingredients for the essential medicines market, but we're also very open to collaborating with companies if they have ingredients in mind that they want to make products with. We're really open to explore the opportunity and really use our technology as a platform to provide solutions not only for our internal projects, but also where there is market opportunity with external partners. So it's really those two models where we are providing access to ingredients that are in short supply, that we can enter the market with ourselves or use collaborators with sales channels already available and use their support to enter the markets with them.
So for some of these kind of rare plant-derived ingredients, you'd think it would make more sense maybe to produce them in plant cell culture, for example, but why does it make more sense to produce them in microbial systems if you can?
I mean, it sounds like a logical step, right, to produce them using the natural plant cells. However, the issue is that these cells are usually growing very slowly, and it's just really hard to have an efficient process. Whereas the E. coli strains that we use are really fast growing. I mean, they're really easy to culture and grow very fast, and are also pretty cheap to culture as well. So by using that as a platform that we can modify in any way that we want, we have this cheap production that we can use for almost any natural product without relying on the slowly growing specific plant cell that's used for that one ingredient.
Let's talk about Fermelanta itself. I understand the company was founded fairly recently, 2022,
Yes.
But you're drawing upon many more years of academic research?
Yes. So the founders of our company, they have been working on engineering bacteria strains for the last 20 years, right? So their goal was really being able to manipulate and controlling on a fundamental level, like how cells, E. coli cells specifically behave. So all of that knowledge now comes in. Even though we're a young company, we can use that as a foundation to really build a platform of E. coli that can produce a lot of useful ingredients for us.